Alrighty, so let's talk about blood. Okay, so in this chapter, we're going to talk about plasma and the cellular elements of blood, how these blood cells are produced, red blood cells. We're going to hit on that, some platelets, and finish off with hemostasis. So when we're looking at blood, right, we know that blood is going to be made up of basically two components: the fluid and the which is going to be like the matrix portion, right? And it's going to also be those formed elements. So the plasma, all right, which is extracellular fluid, right? The fluid matrix of your blood is going to be your plasma, and that's going to make up 92% water, 7% protein, and the remaining 1% is going to be ions, right, such as sodium, potassium, chloride, hydrogen, calcium, things like that. Even organic molecules are going to be in your blood, right, in the plasma portion, glucose, things like that, right? As well as dissolved gases such as oxygen and carbon dioxide. You can have some trace elements in there. You can have iron in your blood, right? You can have vitamins in there. So that's all going to make up the fluid portion of your blood. Now, this type of extracellular fluid, right? So we know we have two types of extracellular fluid. It's going to be your blood, right? Your plasma, specifically, right? That portion of your blood, as well as the interstitial fluid. Now, the plasma is going to be, in a sense, almost a little bit identical to the composition of interstitial fluid. However, we have plasma proteins in your plasma, right? Whereas there's no proteins going to be in that interstitial fluid, right? And so we're going to have those important plasma proteins such as the albumins, the globulins, the fibrinogens, the transferrin proteins, things like that. And what's going to make up your formed elements or your cellular elements, right? It's going to be those cells, right? It's going to be the red blood cells or the RBCs. Red blood cells are also called erythrocytes, right? We're also going to have platelets. Platelets are also called thrombocytes. These platelets, they're going to split off from that megakaryocyte, right? And that's how they get into the blood. And then we have white blood cells, right? These are also called leukocytes, right? Such as your lymphocytes, monocytes, neutrophils, eosinophils, and basophils. And we'll talk about those when we talk about immune, okay? But even going back to the plasma for a second, right? Such as those organic molecules. I know I mentioned glucose, which are, of course, you're going to find proteins, amino acids, lipids, right? Those pools of glucose, free fatty acids, pools of amino acids, they're going to be in your blood too, right? And so those plasma proteins are very, very important, right? Many of them are going to be produced by the liver and they're going to then be secreted into the blood. So albumins, they're going to make up 60% of the plasma proteins. These are going to be the smallest of the plasma proteins. However, they contribute to the osmotic pressure, right, of the plasma. Right, and they also help to transport things such as fatty acid, thyroid hormones, and some steroid hormones. Remember, certain hormones, especially those steroid hormones, those are lipid based, right? Having that cholesterol precursor, so they're not going to easily want to, go in the plasma, which is water based, right? Your plasma is 92% water, and these steroid hormones, if they're made up of non-polar species such as cholesterol, they're not going to want to just remain just freely roaming in the plasma. So they're going to hitch a ride, right, on like albumin, for example, to get around the body. We also have globulin proteins. They're going to make up 35% of the plasma proteins. And there are going to be two types of globulin type of proteins, right? There's going to be those transport globulins that help to transport ions and hormones. And then you're going to have those immunoglobulins, such as your antibodies, right? And they're going to be able to attack pathogens. However, now these immunoglobulins, these are a type of gamma globulins. These are not going to be produced by the liver. These are going to be produced by other specialized cells, your B cells, right? And then you have another type of plasma protein, right? These fibrinogens, right? These are going to be the largest of the plasma proteins. They're going to be the fewest, but these are going to be the largest. And these are going to be very important in blood clotting processes. And they're going to convert to fibrin to form a clot, right? If there's ever a break in your blood vessel. Okay. Now, I do want to hit on just because just the word, let's look at fibrinogen for a second. When you see something with ogen like that, typically is going to be stored and inactive, right? So it's not going to be active. And that's why we are able to convert fibrinogen to fibrin, or even angiotensinogen to angiotensin 1, right? So things that have ogen on the end, like OGEN, right? They're typically going to be inactive form or inactive form of a protein, right? So fibrinogen, angiotensinogen, pepsinogen, right? So things like that, okay? And to get back to the plasma proteins, we have other plasma proteins like prothrombin, transferrin, and things like that too, okay? Now, here I give you just some of the different globulin type of proteins based on their class. So we have alpha 1 globulin proteins such as alpha 1 antitrypsin. You have alpha 2 globulin proteins such as angiotensinogen. We have those beta globulins. They're going to work in, they're going to function as the coagulation factors. And the example would be like transferrin as a beta globulin. And then you have those gamma globulin plasma proteins, and those are going to be like your immunoglobulins or your antibodies. And here just shows you an electrophoresis graph of your plasma proteins. Notice how we have an abundance of albumin, right? In comparison to the other ones, alpha 1, alpha 2, beta, and gamma proteins, right? And so, well, where do these blood cells come from, right? These blood cells, they're going to be produced in your bone marrow, right, through a process called hematopoiesis, right? And so red bone marrow is going to be red because it contains hemoglobin, right? 25% red blood cells, 75% white blood cells, right? Your yellow bone marrow, right? As adults, we contain yellow bone marrow. And that's going to contain adipose cells, right? Inactive, but adipose tissue, right? However, babies do have, or infants, right? They do have a lot of yellow bone marrow too. Now, hematopoiesis is going to be controlled by cytokines, right? And these cytokines that you can see down here, right? Cytokines are simply just peptides or proteins released from one cell that's going to affect the growth or activity of another cell, right? And so we have different cytokines that we see. I'm going to hit on especially these two, erythropoietin and thrombopoietin. So thrombopoietin, right, TPO, is going to be produced in the liver, right? And it's going to influence the growth or the differentiation of megakaryocytes, right? And this is where those thrombocytes are coming from. These are platelets, right? And so it regulates platelet production. Erythrocytes, right? They're going to be stimulated by the hormone erythropoietin, or there's a glycoprotein called erythropoietin. So this erythropoietin, EPO, is produced in the kidneys primarily. However, it regulates red blood cell production. So a stimulus for EPO to be released is low oxygen, right? So if you're in a hypoxic condition, right, your body will produce erythropoietin. Erythropoietin, right, will go to the bone marrow to stimulate the production of red blood cells so we can increase our oxygen carrying capacity. Now, in the bone marrow, right, we mentioned RBCs, or red blood cells, are going to be produced in red bone marrow through a process called erythropoiesis. Because of that glycoprotein erythropoietin, it's also going to be assisted by several cytokines. But like I mentioned, right, the stimulus for EPO synthesis and release is hypoxia, right? So low oxygen levels in the tissues. That's going to turn on the EPO gene and increase EPO synthesis. And then EPO, or erythropoietin, right? It goes to the red blood cells goes to the bone marrow to make more red blood cells, puts more hemoglobin into circulation so we can carry more oxygen, and that takes us out of a hypoxic situation, right? Now, erythropoietin is going to be made on demand and not stored as classical peptide hormones are. And I do want to bring attention just for a second to this megakaryocyte right here. So these platelets, right, as you can see, like these little platelets, let's zoom in a little bit. Well, or not. Let's look right here, right, these little platelets, right? Because of this large megakaryocyte, it can't fit through that endothelial lining, right, of that blood vessel. So it kind of breaks off. And what breaks off, those portions that break off, those membranous sacs, these are those platelets, basically. Okay? That's how platelets get into the blood supply. And you can see how the red blood cells, how they get in there, right? They come from a stem cell, they become ultimately an immature reticulocyte, and then it expels its nucleus, and then it becomes a mature erythrocyte after a few hours. Okay. Now, just simply looking at a complete blood count, it's commonly known as a CBC, is going to provide information, important information about a patient, right? You can look at the mean corpuscular volume, and this is going to be the average volume of one red blood cell. You can look at the amount of hemoglobin per red blood cell, right? And you can look at the amount of hemoglobin per volume of one red blood cell. So there are certain tests where we can test for, how many red blood cells are there, how much hemoglobin is there, and the hemoglobin is really going to be important to tell you about how much oxygen your body can carry. Okay. Now, a hematocrit is very important. This is a test, right, that shows or gives you the ratio of red blood cells to plasma, right? And it's going to be expressed as a percentage. So it's the percentage of red blood cells in your blood, right? And it's going to tell you about your oxygen carrying capacity of your blood, okay? So for males, their hematocrit will show 40 to 54% percentage of red blood cells in the plasma, right? And as females, it would be 37 to 47%, give or take, okay? Now, red blood cells, right, when we're just simply looking at their anatomy, right, they look like these biconcave discs, almost like jelly donuts without the jelly part in the middle, right? And it's because if we look at these red blood cells, a mature red blood cell is going to lack a nucleus, right? And however, because they're shaped so specifically, right, their morphology can definitely provide clues to the presence of a disease, right? And so what happens is aged and damaged erythrocytes, they're going to be broken down by the spleen under normal conditions, and I'll show you that in the image in just a few too, right? But you can see the cytoskeleton of these erythrocytes. They're going to create unique shapes, and that what gives this red blood cell its shape, right? And these red blood cells, these are basically like membranous sacs. They don't have a nucleus, right? They don't have like a mitochondria. They're not going to, if you remember, mitochondria is going to be the organelle for oxidative phosphorylation. And so they use oxygen, right, to create ATP. And if you're using the oxygen that this red blood cell is carrying, right, the whole purpose of red blood cells really is to carry oxygen, right, around the body. And so if you're going to use that oxygen, the oxygen won't be able to get to the tissues, right? So we don't want to have a large amounts of proteins in here. Unfortunately, because we don't have a nucleus, it's not able to, create proteins and even repair itself. So when it's damaged, right? And see here, you can see down here, this image right here, right? You can see an erythrocyte or a red blood cell placed in different solutions, different mediums, right? So this first one, right? A hypertonic solution, right? Erythrocyte is going to be placed in a hypertonic solution. However, that rigid cytoskeleton remains intact, creating like the spiky appearance. And these cells, they actually crenate, right? Versus if you put a erythrocyte in a hypotonic solution, right? They're going to swell up, right? And they're going to lose their characteristic biconcave disc shape, and they're going to become like a ball or blob, right? I think of hypo or hippo, right? And then we'll talk about sickle cell disease too, that sickled cell shape, okay? Now, I mentioned red blood cells, they don't live very long, especially because they don't have the nucleus, right? So they live for about 120 days. Those older red blood cells, they're going to rupture and get phagocytized in the spleen, right? The amino acids from the globulin protein, they're going to be incorporated into new proteins, and some of the iron, the globular proteins, the globular regions of the heme group, I should say, is going to be incorporated into new proteins, and some of the iron from the heme groups are going to be reused, right? So recycled in these new heme groups. So as you can see, iron is really important for the synthesis of hemoglobin, right? And hemoglobin is going to be that oxygen carrying protein that's going to be located in the red blood cells. So iron must be ingested from our diet, right? As we ingest iron from our diet, it's going to be absorbed by active transport over the epithelial lining, right, of your small intestine. And so we have these proteins, right? We have transferrin, right? Which is a protein that's going to be in your plasma, and it helps to transport iron in the plasma. So to transport that iron, take it to the bone marrow. The bone marrow is going to use that iron to make hemoglobin as part of red blood cell synthesis, right? And if we have too much iron, then it's going to store that iron in the liver, okay? But the iron that gets transported into the bone marrow is going to be used to make hemoglobin to make red blood cells. Now, after 120 days or so, right, as that red blood cell has now been, is old, right, the spleen is going to destroy old red blood cells and convert that hemoglobin group that's within that red blood cell is going to convert that hemoglobin to bilirubin, right? And so that bilirubin will then get transported to the liver, where it's going to get conjugated, and then it's going to be produced into its bile, and then that bile will then be stored in the liver, right? I'm sorry, produced in the liver, and then it's going to be stored in the gallbladder, and then it's going to be very, very important because we use that bile to be secreted into that duodenum so we can emulsify fats. And so we'll talk about that during the digestive lecture, though, right? And however, if you have a large amounts of bilirubin, right, we'll excrete it in our urine and our feces, right? And so, but remember that the remnants from these heme groups, right, they get converted to bilirubin, okay? And it's that bilirubin metabolite that's going to be excreted in your feces and your urine that's going to contribute to that yellow color, right? Now, if that bilirubin is not conjugated, right? So say, for example, you have liver failure, right? And you're not able to conjugate that bilirubin, right? That bilirubin will increase in the blood, and then that can lead to jaundice, okay? And so typically when you have an individual with jaundice, especially like a newborn baby, right? Newborn baby with jaundice, they'll just put them under light, right? So the light can help them synthesize and remove some of that bilirubin out of the body, and then they'll remove it in their fecal matter, and then you'll see their fecal matter being like extremely yellow, or extremely dark, I should say, okay? Now, hemoglobin synthesis require iron that we just mentioned, right? So hemoglobin plays an important role in oxygen transport, right? And so when we're looking at hemoglobin, right, there's 4 heme groups. So you can see 1, 2, 3, 4. So 4 heme groups. There's going to be several isoforms of hemoglobin. You have like adult hemoglobin, you have the fetal hemoglobin. And so the adult hemoglobin, which you see right here, we have 2 alpha subunits and 2 beta subunits, right? So 2 alpha and 2 beta chains that's going to make up this globular protein, right? Fetal hemoglobin, they have 2 alpha and 2 gamma chains. That's going to make up their hemoglobin, okay? And so this is that heme group right here that's going to be in the center of those polypeptide chains, right? And so the heme is going to be a porphyrin ring with an iron atom in the center. And this is where 70% of your body's iron is going to be found at, right? And of course, iron comes from your diet, mainly from like red meats, beans, spinach, and really just iron fortified foods, such as iron fortified breads. But that iron, again, is going to be transported in the blood by transferrin, or transferrin, I should say, right? And iron is going to be taken up in the bone marrow. Excess iron, again, is going to be stored in the liver by ferritin. Okay. Now, I do want to mention, because excess iron, like so too much iron in the body, now that can be toxic, right? And it can be poisoning sometimes to children when they ingest, too many vitamin pills that contains iron. And so initial symptoms of that iron toxicity are like, gastrointestinal pain, cramping, internal bleeding, which, occurs as iron, because too much iron, it's going to corrode that digestive epithelium, right? And so, of course, subsequent problems will include like liver failure, which, of course, can be fatal, right? Now, there are some disorders, right, when it comes to our decreased oxygen transport, right? So our red blood cells, are important in transporting oxygen. Now, anemia, right, is where we have our hemoglobin content is just too low, right? So ultimately, we have a low oxygen carrying capacity. So different causes can happen or can cause anemia, right? You can have accelerated red blood cell loss, right? And this is where you're bleeding out, right? So you're hemorrhaging, you're bleeding out, you lose too many cells, right? So if you lose too many red blood cells, you lose too many or too much hemoglobin. Thus, you're reducing your oxygen carrying capacity, right? So you're losing more red blood cells than you normally should. There could be hemolytic anemias, right? And this is where your red blood cells or your cells are just simply getting destroyed quicker and rupturing quicker than they normally would, right? You can have membrane defects such as hereditary spherocytosis. And so with this issue, instead of having a biconcave shape, these red blood cells, they're more spherical. And so the membrane gets damaged a little bit quicker, and so they get destroyed quicker, right? And then you have like sickle cell diseases and iron deficiency anemia as well. And we'll talk about sickle cell anemia in a minute, though. But you can also have acquired anemias, and so such as like malaria or parasitic infections, they cause anemia. Certain drugs and certain autoimmune reactions also cause anemia. Another cause of anemia is decreased red blood cell production, right? So you're not making the normal number of red blood cells at a rate you should. So whether you have like defective red blood cells or hemoglobin synthesis in the bone marrow just decreased, right? So you have aplastic anemia. Aplastic anemia can be caused by certain drugs or radiation, and so aplastic anemia is where your bone marrow is impacted. So, for example, like radiation is destroying your bone marrow, and it causes it not to work properly, and you can't now create red blood cells like normal, right? So you have a low red blood cell count, thus low hemoglobin, thus low oxygen carrying capacity in the body, right? So iron deficiency anemia, if you're not taking in enough iron, because we know iron is important for hemoglobin synthesis, and hemoglobin synthesis is important for red blood cells, right? Issues with vitamin B12 deficiency, right? And so this right here with vitamin B12 deficiency, so it's not that you're not taking in enough vitamin B12. It's the problem is you're not able to absorb it. So we have parietal cells that's in our stomach, and these parietal cells, they release intrinsic factor. And intrinsic factor is required for the absorption of vitamin B12 in your intestines. So if you don't have it, you can't absorb vitamin B12. And vitamin B12 is very, very important in DNA synthesis. So vitamin B12 deficiencies, this can lead to pernicious anemia. Okay. So sickle cell anemia, right? This is going to result from abnormal hemoglobin molecules. And so basically it's a genetic defect in which glutamate, which is that sixth amino acid in that 146 amino acid beta chain of hemoglobin, that glutamate is replaced by a valine, right? And so one amino acid change, right, can produce these results that you see right here. Now remember, DNA gets transcribed into RNA, RNA gets translated to protein, but if you have the wrong nucleotide, right, that's going to bring up the wrong amino acid ultimately. As transfer RNA brings in the correct amino acid, it's going to be bringing in a valine instead of a glutamate, right? And so that right there will result in an abnormal hemoglobin. And so that abnormal hemoglobin crystallizes when it gives up its oxygen, and that crystallization pulls the red blood cell into a sickled shape like a crescent moon. And so these sickled cells, they become tangled with other sickled cells as they pass through the smallest blood vessels like capillaries, and they cause these cells to basically jam up and block blood flow to the tissues. Excuse me. And that blockage creates tissue death, tissue damage, and pain, right? From not receiving oxygen, from being hypoxic. Now, platelets, right? Platelets are these cell fragments from the megakaryocyte. These are going to be smaller than red blood cells. They will contain a mitochondria. They also contain smooth endoplasmic reticulum and granules, right? Such as like the membrane-bound vesicles containing cytokines and growth factors. They're not going to live that long either. They're going to have a 10-day lifespan, but they are going to be important in stopping blood loss. They're going to be important in immunity and inflammation, right? So these platelets, they're going to function to release like these chemicals that are going to be important for clotting if primary hemostasis is not enough, right? So we want to form that temporary platelet plug, right, to those injured areas, right? And so that will basically stop, bleeding, and then we can initiate. It'll stop bleeding out of that vessel, right? That injured blood vessel, and then we can initiate the clotting process. Okay, and this just looks at hemostasis, right? Hemostasis is blood clotting. We're trying to prevent blood loss, right, from that damaged vessel. So what we're going to do first is we're going to have a vascular spasm, right? Or we're going to vasoconstrict. So we're going to reduce the amount of blood that's coming to that area, right? What happens is because that blood vessel has broken, right, or there's a break in the blood vessel, as you can see, like right here, right, that collagen is going to be exposed, right? And because that collagen is exposed, it's going to bind and activate platelets. So these platelets are going to release these factors, and these factors are going to attract more platelets to this area. So these platelets are going to aggregate to this area, and they're going to create a platelet plug that you can see right here, right? That's going to be that platelet plug, okay? And so then we're going to be able to so we have that temporary hemostasis, right? Because we have that platelet plug, right? Then we're going to be able to reinforce that platelet plug, right? So the platelets, they're going to stick to that collagen. They're going to release that prothrombin activator. That prothrombin activator is going to activate thrombin, right? So prothrombin is going to be one of those proteins, one of those plasma proteins. Let's go to this slide because it gets it better, right? So prothrombin is, let me write it over here, prothrombin, right? It's made in the liver with the help of vitamin K, right? That's why vitamin K is really important in this factor. So the platelets, they're going to release prothrombin activator. So prothrombin activator, right? Prothrombin activator, these platelets are going to produce that. This prothrombin activator is going to provide the pathway for prothrombin to get converted into thrombin, right? And thrombin is basically like molecular scissors that cut proteins. And so thrombin will then act to convert fibrinogen, right? So it's going to cut it, right? So converting fibrinogen to fibrin, right? And fibrin is going to be really important because fibrin are basically like these protein threads. And so these protein threads, this fibrin is now able to reinforce the clot. And then we have like the remodeling process that is going to occur where we're going to be able to fix the blood vessel wall by putting in connective tissue, and that's going to be help mediated by this fibrin. And then when we want the fibrin to go away, we'll go through a process called fibrinolysis to lyse to thrombin when that connective tissue is able to repair itself. Okay, these are just the two pathways, the coagulation, how coagulation converts a platelet plug into a clot, right? We'll talk about this more in lecture, but there's the intrinsic pathway known as the contact activation pathway. That's going to be like from within the blood vessel. And then we have the extrinsic pathway known as the cell injury pathway. That's going to be known as like outside the cell to control this clotting formation. Okay, but of course the common pathway between them is going to have that thrombin converting that fibrinogen into fibrin, and fibrin becomes part of that clot. And like we mentioned before, fibrin is broken down by an enzyme plasmin, right? That's an inactive plasminogen activated to plasmin by a tissue plasminogen activator. And fibrinolysis is important because we're going to be able to remove or break down that fibrin ultimately at the end, right? And so these anticoagulants, they're going to prevent coagulation, right? So thrombus is going to be a blood clot that adheres to an undamaged blood vessel wall, right? So there's going to be two mechanisms that's going to limit the extent of our clotting process so it's not going too far, right? So inhibition of platelet adhesion and inhibition of the coagulation cascade and fibrin production, right? So we also have endogenous anticoagulants such as heparin, antithrombin 3, protein C, things like that. And I do want to mention like other like several diseases in which one or some of these factors in this coagulation cascade is going to be defective or lacking. That is so known as hemophilia. Okay, and that was this chapter. Okay.